people signed for yesterday. So if you were present if you were present today and yesterday, sign for both. If you, well, you know what to do. This is for you. Okay. So while people are also joining, so there, okay. So there, before I start and I ask if there are questions. So one announcement: we have there is a TA for the class who is saying no with red, but she's. So is Roa sitting? Yeah, you can stand up. So everyone. So if you have questions, etc., you can also ask her, and she's happy to help with this. Okay. So I think it's a good moment to start. So before I start, any question? Yes. Mm-hmm. Okay, so the question is, I can take the slide actually, going back to that. So the question is, when we looked at the very first slide I showed this tree of life, and I showed that there are many organisms which are the one with the red dots that we know that they exist because their DNA was reconstructed from DNA collected in the environment. And the question is why we are not able to grow them in the lab. So I mean, if we knew the exact answer, it means that we would be able to grow them, right? If we know the conditions that. The point is that growing something in the lab requires that you know something about the organism. And I mean, not the only way, but the main way we know something about organism is if we grow it in the lab, isolate, sequence, and perform experiments. So the point is, it's a bit trial and error. So when people try to isolate bugs from the environment, what they do is that they try conditions that sort of are very permissive, so they are at their very optimal temperature, or that they are like close to their optimal temperature, they have plenty of nutrients, etc. The point is that there might be some micronutrients or something which you don't put that is necessary. And sometimes the other point is that these organisms are extremely slow growing. So for instance, the most abundant organism on planet, so the organism with the largest number of individuals is a bacterium, of which you know the taxon is called SAR11. And it's the most abundant, so it lives on the surface of the ocean. And there are 10 to the 24 individuals, so more than Avogadro number of individuals on Earth. And it's very hard to grow, extremely hard. So for one species, people were able to grow it, but it's painfully hard to grow it, and it reproduces extremely slowly. So if something reproduces at the optimal condition every 24 hours, if you have a bad condition, you have to wait months, and then how do you know that this thing is dead or not? So that's the answer. Other questions? Okay. So we can start today. So I actually had a question or a comment, and I want to, about something that I might have misrepresented, so about the role of enzymes. So just to stress something that I thought was obvious, but is not obvious. So the main role of enzymes is that they decrease this energy barrier for a reaction to occur. So basically, they speed up a reaction. So I sort of misrepresented this because when I was plotting the derivative of the products as a function of the reactant, I say that in presence of enzymes, you have something like this. And sort of in the previous plot, I showed what happens in absence of enzymes. And I showed something like this, which is linear. What I should have shown yesterday was something like this. okay? So that in absence of enzymes, the rate at which this reaction occurs is extremely, extremely slow, okay? While in presence of enzymes that, if enzymes are very abundant, that rate is governed by the catalysis rate, which is much higher than the rate of spontaneous reaction, okay? Is this clear? Good. Very good. Okay, so we arrived at this point yesterday where I said the kind of experiment you can do in microbiology, and I said one thing that you can do if you are able to do it is to grow bacteria, and that's the way you do most of experiments in microbiology. So you have some medium, you put some nutrient, some source of energy, and you inoculate some bacteria that you know they are like what species they are, and then you have this flask, and you want to shake it perhaps because you want to be that oxygen is there, and then what you do is that you wait. And for instance, what you can do is that you can see that your medium, your flask, it becomes more and more turbid because things are growing. And if you look at how turbid is the medium, so basically as a proxy of how many cells there are, what you see is that at the beginning things don't grow, then they start growing, and then they sort of saturate, okay? So the idea is at the beginning, these things arrive in this new environment, they are still sleeping, so they have to wake up. Then they wake up, they start to reproduce, but then there is a point where nutrients get depleted in the environment, or they release some toxin which slows them growth. There is something that limits their growth, and then they sort of stop, and then they stop growing. And then, there is another thing that happen, that if you wait a while, then it starts going down because cells start dying, if you track the number of cells, okay? And what I said is, okay, so you can grow these things, and you want to understand how they function, what keeps them alive, what they do when they are alive. And what you can do is that you can take a sample from your flask after they grew for some time, and what you can do is that you can look at under the microscope and see, for instance, how big they are, what's their size, some properties of individual cells, or you can do some if you want, biochemical extractions and measure what is their composition, how many proteins there are, how much RNA is there, or you can also characterize individual proteins, so you can measure really what is the internal composition. If you do this, you measure the internal composition of the bulk, so of all the cells averaged together, okay? And so you can imagine that you know from yesterday and probably from before, more or less what are the functions that a cell, that cells have to do to be alive. Suppose that you want to describe this function. You want to learn, say something more about this function, be a little bit more precise about this function work. And my first question is, so suppose, for instance, that you want to measure how regulation of a given protein occurs. Right? Now, the point is, the first question that you have to ask yourself is, when I take this sample and measure cells, right, measure the property of the culture, when should I measure it, right? You can say, I could measure it here, 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 okay? And so this is the question I asked you yesterday. And so now you have to answer, okay? So we can do a poll, okay? Who thinks that you should measure here at the beginning? One, okay. Who thinks that you should measure here? Okay, let's say 27%. Who thinks you measure here? Okay. I don't know because I don't know the total actually. So it's, let's say it was 1/3 of before, so 9%. How many here? Okay, about 12%. Okay? And that doesn't sum to 100 because people didn't vote, some people. Okay, so there are some different opinions. So there one person that thought here. Why? Sorry.
I think we should measure whenever we can.
Whenever you can, yeah, but you have to make a choice, right?
In the middle.
In the middle, because, okay, so you are, okay. So then there were people that said you have to measure here, in the exponential phase. Why? There is 27% of you who said that. So why in the exponential part? Hmm? Okay, if you want to measure the rate of growth, yes, but perhaps I say, do you want to measure the rate at which growth decreases, and so you should measure it. But if you want to see how, I mean, this is not, just to clarify the question is not about when is most interesting, okay? The question is, when is easier? In the end? So do you want to retake the vote after I say this? Okay, so who thinks we should measure is easier here?
What do you mean by easier?
That whatever results you find, it's easier to interpret and to model, if you want.
But can you make an example of a kind of measure? Like
You measure, for instance, you want to measure how, let's say that you have a regulation like arginine, like before. You want to understand what are the parameters of this regulation. What regulates what, and you can perform different experiments. Or you want to measure how much, I don't know, the amount of DNA that you have depends on the nutrients you put.
So it's what is the most representative, in a sense.
It's not the most representative. It's what do you think makes it easier than to, I mean, it's a fuzzy question on purpose, but what makes it easier? When would you do it if you had to do it? If you wanted to do an experiment, and let's say you want to know, let's say the DNA, you want to know how much the concentration of DNA, how much DNA you have, depends on the nutrient you put.
Are you measuring time here as well?
You can measure time, yeah, I mean, you can measure also at different times, but.
Then probably the third phase is the most easier way to interpret, and then like you can
Okay.
get an average rate from the beginning till the end. I mean, I think now the third phase is like,
Why do you think the third?
well, you can present when the system has saturated, and probably it's the most equilibrated
Okay.
phase in the system, and then like you can predict the rate because you have the time.
Okay, so your argument is it's a very interesting one, is you should measure at stationarity here, because it's called stationary in some sense. And since it's called stationary, the properties of the cells will be stationary somehow, will not depend on an history, okay? It's a very good argument. Other points? Yes?
I choose the late exponential.
Okay.
Because, okay, in phase one, it's too fast to measure all these quantities because it is a log scale. So exponential can grow much faster than in phase two. I think that in stationary phase could sorry, in second phase can be we can find more interesting properties how to grow because in stationary phase maybe the resources or in growth medium can be done. In late exponential, we can see the growth maybe similar like in exponential path before and now.
Okay, so basically you say is this one because here you have properties both of this one and of this one. Okay? Yes, there were two people there.
I think it's the exponential phase because there you have the conditions which are most similar to a natural environment, and
Okay, so this is also interesting. So you said the exponential because this is where typically you expect, it's sort of the natural environment for,
Mostly they are happy, they are not feeling the effect of the small environment you put them in,
okay,
which is what happens in the stationary phase, you may
okay,
have limited nutrients.
okay,
And also, for some species, I think you have collective effects, like they help each other, and so you don't want to be in the lag phase because they are too few and they can't
okay,
cooperate well.
okay, okay,
So in the middle.
in the middle, okay,
I think the saturation in the third part will affect the activity of the materials or the population, and also we want to a constant population of population in our system to get correct information.
okay. Why, in what sense correct?
What?
In what sense correct?
Like this is stable to analyze and
Okay.
measurement.
Okay. Okay, so it's similar to the first argument. Yes.
I want to say like this exponential phase, like we have like two types of growth. There is slow and the fast growth. So imagine like the growth is so fast. There are so many information are going to go because of how much the change occurs.
Mm-hmm.
So I would really like the second phase because at that phase you have like all the growth almost kind of converges to the
I'm not sure in what sense converge. Here, you mean? So just to clarify, this inset here, if it was confusing, this represents what you expect in two different conditions. I didn't say that. So for instance, here you give a nutrient which doesn't support fast growth, and here you give like some nutrients which supports fast growth. Or here it's optimal temperature, here it's slower. Okay? So it's the same. By the way, if during COVID you had like baking yeast, the dynamics is exactly this one, right? When you have your sourdough and you are adding flour, what you see is that at the beginning nothing happens, then it goes very fast, and then it goes here, and then if you forget about it, it goes down. If you put it in the fridge, it's like here. If you put it outside, it's like here. Okay? Just to close the parenthesis for baking friends. Yes? OD is here, for the context, you can imagine is total biomass, how many individuals there are.
Okay, so later is better because there's more, the population is higher.
Okay, because okay. So I think we collected a group of answers, and I mean, as I said, the question is a bit, was on purpose a bit fuzzy also because, I mean, study physiology, it's a very broad question, so when it's easier, so also strongly depends on the question. Of course, if I ask questions that are specifically related to how much time does it take to a cell to grow, I want to study it here. Right? But in general, I think that there is a good argument, which is important to keep in mind, of why the easier condition to study is the exponential phase. Okay? And very interestingly, the reason why the exponential phase is easier is because it's where the property of a cell are stationary. Okay, so exactly for the reason you give, but it's here. And interestingly, this is often criticized as a condition exactly for the condition he gave about exponential because it's not natural. Because in the environment, it's rare that cells experience long stretches of exponential growth. Okay? So what I want to show you now is to give you an intuition about, yeah? I'll explain now with a simple model why exponential growth. And so then I perhaps give again the intuition. So imagine that we have our colony, okay? And our colony is growing, okay? So our colony is growing, and what we can imagine to measure is the change in time of the Ni, which is the total, let's say, number of proteins of type I, or whatever molecular species you are interested in. This could be proteins, this could be whatever, of type I, right? Any molecular species you care about in a cell, okay? How does this change on time? Well, this will change in time. It will have some dynamics, okay? It's some function f, which will depend on, in principle, all the molecular species that you have, okay? And it will depend on properties of the environment, okay? So if you want, the change of the number of molecules of type I depends on all the molecules you have in your culture and what is the environment, okay? Do you agree? Okay. Now, here there is first complication. What is the environment? Okay. Now, in principle, you can imagine that the environment, let's say, the alpha property of the environment, which could be something about the environment, could be pH, could be amount of nutrient, could be whatever, also depends on the growth of the cells, etc., etc. Right? If we have this dependency, what we are saying is that as cells they grow, they change the environment, and the environment change the composition of cells and their growth. And so we will start to talk about ecology, and we don't want to talk about ecology here. Okay? I'm sorry for the ecologist friends. Okay? Including myself. So we don't want to care about this. Okay? Now we want to study physiology. We want the condition to be fixed. So what we have here is that we assume that this is constant, does not depend on time. So in principle, it depends on time, but we assume that for us is constant. Okay? Now, opening a parenthesis, which is not a parenthesis. What's happening here, the reason why this goes down, okay, intuitively, I've not shown it in equation, but intuitively, the reason why this goes down is because the environment is changing, because the nutrients are running out. So if I measure how many nutrients there are in the environment, what I see is that this goes down, right? And ecologists know this very well. Any form of growth limitation, it's due to some sort of interactions, either with conspecific or other individuals. Okay? So this is the first point. Okay? The second point is that we can imagine, okay, we are tracking the sort of number of molecules of a given type inside our colony. What we can track is we can track also the total number of molecules, or we could track their mass. So each molecule of type I has a given mass. So this will be the dry mass of a cell. So if you remove water, what will be the dry mass of your colony? Or we can imagine to track volume. Okay? You can take all the cells and you look how much volume they occupy. Okay? Now, what we can imagine is that, let's say that if we focus on volume, so we have this. Let's say we also have that dV T will be some function of what? Some function of all the n of the volume and of the environment. Okay? Now, any questions about this? Yes?
I'm confused. Why are we tracking the number of molecules and not
And not?
organisms? Organisms.
Well, organisms, you can imagine that it's related to volume, right? You can imagine the number, you can imagine that all these things are proportional, In a sense, it's an assumption. But
Volume is the total volume of the...
you take, yes, we are assuming that individual cells have more or less the same size. And so the number of cells, let's say B, the total number of cells, it's another things you could track, right? But we are assuming that all these things are proportional to each other. Okay? So I'll do it with volume, but you can do exactly the same things with any of these other properties. Yes?
Joining N and V as density.
Yeah, you can. This is what we're going to do in a second. Okay? Is this clear? Now, an assumption, if you want, that we can make, which is a simplifying assumption, is that if we have our colony growing, and this colony is, let's say, big enough, they don't interact, they don't consume nutrients, and I cut it in two, right? And I study what is the number of molecules in part A, and what is the number of molecules in part B. Okay? The sort of, I can neglect the fact that there is, this interface doesn't matter. So basically everything is extensive if you want. So that dNi A plus, so I can write dNi A dt will be Fi N A E and dNi B dt Fi N B E. Okay? But this distinction is arbitrary, right? So if I write d NiA plus NiB dt, this should be equal to Fi Na plus Nb E. Okay? So this means that if I want to have this property to hold, what I can introduce like dN over V, so concentrations, okay? And I can rewrite the equations for dN dt. If you want, I can write my function Fi of N E as some function of the concentration C E times V. Okay? Yes. Sorry? What is there? I separating. No, this is arbitrary. It's like you have your colony and you say, if I now track only this part, and I track only that part, I can look at the growth of this part and they don't interact, right? So they don't interact, so if I look at only that part, it will grow this way. And the other part, it will grow this way. But since this is completely arbitrary, this condition has to apply. And so the only way I have, if I want to express it with V, is this one. Okay? Yes?
What you meant here is that N is an extensive parameter.
Yes, exactly. So that if you want all, that's exactly right, that B, V, N tot, M, and each Ni are extensive parameters, right? So I have extensive parameters, so I can express the change of these extensive parameters as something extensive. And I express something extensive, I have an extensive quantity times a function of all the intensive variables. Okay? Now, similarly, for this function here, what it means is that G of N, V, and E is equal to small g C E times B. Okay? Exactly for the same property. Okay? So now I have an equation for V, which tells dv dt is equal to dc E times V. And then I can write, well, in principle I could write the equation for n, but since I'm expressing everything in terms of concentration, it's better to write an equation for concentrations. So I can write the equation for dci dt, which is equal to ni over V, which becomes 1 over V dni over dt minus ni over p squared dv dt. Okay? If you want, I can write as 1 over v dni dt minus ci 1 over v dv dt. Okay? So if I write here the equation for concentration, I get that the change in concentration of the molecular species I is equal to what? To the derivative of ni dt, which is this term, divided by v. So it's this term divided by v. So it's fi times c of ce minus ci times 1 over v dv dt. Okay? 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 Now, before I comment what we found, questions about the sort of mathematical steps. Yes?
Could you please repeat that? Why you assume that function f and g are just simply proportional to the volume?
Because of this condition here. Because they have to, because if this has to be equal to the sum of these two, right? And I'm assuming that the biomass, the volume, the total number of proteins, the mass, it's like this. I need that this quantity here, it's linear in the sum. Right? Okay. And so basically what is, I mean, if the volume scales, it means that the concentration here and the concentration here is the same. Right? So the idea is that the concentrations here is the same. And so when I sum them, I sum the concentration times the volume, so this is equal to the volume. So basically, this expression here implies that if I write Fi Na plus Nb given E, I can always write this as Fi C times Va plus Vb, right? Because I'm assuming that the concentration, the intensive quantity are the same. And here, it's equivalent to, if I write this, I mean, if I want this to be equal to Fi of C of Va E plus, so this is the condition I want. Okay? What I need is that these that these and these are linear in VA. Okay? Other question about, yes? The function g is telling me how, this function here is telling me how the volume change as a function of all, so how my colony is growing. Exactly. Yes. Yes. If you want this, yeah, for the total total amount. Okay. They are not independent, but yeah, sort of. Yeah. Okay. So I have these two equations. Other questions about what we found? Okay. Now, question for you. Do you see why exponential growth is the easiest? So what does it correspond to exponential growth? So imagine that you have this equation, okay?
Because you can, if you put the v on the other part, you can write ln of the.
Yes, so what is exponential growth? Exponential growth is dv dt equal to some lambda v, okay? This is exponential growth. Right? So exponential growth, if you want to say it on one hand, exponential growth corresponds to the case where the rate of growth is linear in the volume, so this quantity is constant, this lambda is constant. So exponential growth corresponds to this quantity being constant. This quantity being constant, it means that the concentrations of internal molecules are not changing, okay? They're not changing over time. If they were changing over time, this will change. This quantity will depend on time, and you will not have exponential growth. Okay? So exponential growth corresponds to the case where this is constant. And so if you look at this equation, it corresponds to the case where this is equal to 0. So to the fixed points of this equation. To say this in the other way, if you have this dynamical system, so you see that this is independent of the volume, right? This is like a purely intensive variable. If you have this, it's a dynamical system. So the first things you do when you have a dynamical system, for some of you back of a few months, for others of you back of 1 year, for others, I don't know. But what do you do with this? The first things you do is fixed points, right? You calculate the fixed points. So you try to find the solution where dci is equal to 0, okay? So if you have this solution here, you have a solution where fi of c of e equal star to c star I g of c star e. So you have a fixed point. But if you have a fixed point, it means that this quantity here, it's evaluated in a fixed point. So it's a constant. And so it means that you are growing that you are in exponential growth. Okay? So the point is, just to repeat it once more, if you are, how do we interpret this growth curve in terms of what we saw over there? Okay? So if you are here, I sort of told you what's happening. If you are here, both C, and not just C, but also E are changing, right? So this is changing, all the, this E is changing, these C are changing as a response to E, because the cell is adapting to the fact that nutrients are getting depleted. And so since these two are changing, this quantity here, so the rate of growth, is not constant. Okay? Is decreasing, right? You see, this is the law. I should have made a plot next to it, but I can do it now. So over there we are plotting, let's say, we are plotting the log of volume. Okay, where volume here we mean the size of the colony. Okay, could be the log of the biomass. And what we see is something like this, right? Okay? If you plot d log of v over dt, which is equal to 1 over v dv dt, okay? So the growth rate, which is equal to g of c of e, what would you see? At the beginning, this derivative, the derivative over time of this quantity, is very small. It's actually close to 0. So this is close to 0. Then it goes up. It goes to some stationary value, to some value here, which is sort of a plateau. And then it starts decreasing and goes back to 0. Okay? So what's happening is at the beginning, let's start at the end. Nutrients, so if we track nutrients, let's say, which are E in our case are nutrients, they are present and then they are going down. So nutrients are changing because they are depleted, and so what's happening is that the cell is adapting to this change of nutrients, so the concentrations are changing, and in response, the growth rate is changing. This function here is changing. Okay? Conversely, at the beginning, there are nutrients, but the cell is not at steady state. It started, we took this cell from another culture, so it started with some different initial condition of these values of C. Okay? And so at the beginning, this dynamic is not in stationarity, it's not in steady state. So the concentrations are changing, right? Since the concentrations are changing, our function G is changing, okay? So at the beginning, the value of G is very small, right? This is also G of C is small. And then it's approaching the fixed point of that equation. It's constant, and then it goes down, okay? Is it clear? I see for some people, they seem convinced. Other, I see signs of perplexity. Yeah?
So the exponential phase is the most robust to external perturbation?
No, in the sense that, sorry for the no like this, in the sense that if you change something in the environment, you change exponential growth. Right? So if now I'm here and I change something, I increase the temperature, then what happens is that these external conditions change. Right? And so now these external conditions change. So for the new condition, the cells is not anymore at steady state. The concentrations are not anymore at steady state, so the cell has to adapt again. Okay? So for instance, if you have an abrupt change of condition, let's say that we look at d log v over t, right? We are in steady exponential growth, and then what we do is that we have a switch of temperature, right? What happens is that you might see something like this. Okay? Because the cell, or could be something like this, I don't know. But what happens is that the cell has to respond, right? So changes the internal dynamics, changing this C. As a consequence, at the beginning, you are in a new condition, right? But the concentration is not at the steady state. Okay? Is it clear? Is it not clear? Questions about this? Are you convinced that you want exponential growth? Yeah?
I have a question with minusing or yes. It doesn't make yes, but I don't find the sense because G has to be.
Okay. This was thank you, because I was about to forget this point, but it's a comment I want to make. So what does this equation mean? You have two terms. So you have a term which is something related to the growth of the molecule of type I, and then you have minus Ci times something, right? So if you are in exponential growth, this quantity here, it's the growth rate. So this term, it's basically dilution by growth. So suppose that you immediately, in some way, you stop producing a molecule. So this one becomes 0. Now, your cells are growing. So since they are growing, the concentration, the volume is increasing. So the concentration of that molecule is decreasing. Because cells are growing, you are not producing that molecules. So, right? So this is dilution by growth. Okay?
I'm a little confused. Is V the volume of single cell or
No, the colony. The colony. Was here. So it's for us, the volume, the biomass, the mass of cell, the dry mass of cell. So you take your colony, okay? Let's say you can do different things. So one thing you can do is you take a sample of this colony, you remove water, and you wait. This is dry mass. Another thing you can do is you look at this colony, you shine light through it, and you look how much light passes. That's a measure of turbidity, which is a proxy of how many cells there are. Okay? These are equivalent. Volume, you say, I know what is the density of cell, so I convert the mass to volumes. Right? So suppose that you were taking all the cells together, and you had a blob, that's the volume. Okay? If you want is the internal volume. It's convenient because then you talk about concentrations. Okay? But yeah. Which it's because like concentration, it's what determines the rate of reactions, etc. Yeah. Yeah, okay.
What is the physical interpretation of the third diagram?
The third diagram, it's the nutrients that are depleted, for instance. This is not very important for us, but this is why, after you are in exponential growth, why this starts to decrease. It's because nutrients are getting depleted, okay? And so in our language here, what's happening is that this E is not constant anymore, okay? You will see this in a couple of weeks, I think, in the ecology part.
E was a constant in the exponential phase, yes?
Yeah, if E is not a constant, you immediately see that unless somehow you adapt very, very rapidly, but that's impossible, this will depend on time. So if this depends on time, it means that this depends on time, so it means that you are not in exponential phase. Okay? So the exponential phase, it's like easy, makes things easy, because if you are in exponential phase, it means that the external conditions, I mean, it's both a condition, something that tells you about what's happening, and something that you want to seek for, because then the situation is simpler. So if you are in exponential phase, it means that the external conditions are not changing. And it also means that if you spend a long enough time in exponential phase, so these equilibrate, right? You are really in exponential phase here. It means that the concentration of the internal composition is at the fixed point. So it means, in some sense, that both the physiology and the ecology are stationary. Okay? That you don't have to care about ecology, about interactions, and with the environment. I mean, the environment is fixed, so you don't have to care about how the growth modifies the environment, and that the internal state of cells is the same. Okay? So there is a very nice quote by one of the fathers of modern physiology, which tells you that says the difference between exponential phase and balanced growth. So balanced growth is this one. Sorry, balanced growth is to say basically this argument here is the same difference between watching apple falling and thinking of gravity, right? So it's the same phenomenon, but this one tells you something deeper about what's happening when you are in exponential growth. Question?
I'm a little confused. What is the difference between CI and C? As I can see, CI is a function of time. The second equation is
Here?
yeah,
This is like I'm writing the equation, this bad habit, but I write the equation for component I, and this is like a function that depends on I of all the concentrations, minus the concentration of I times G.
This is no, I mean, I is a function of C and E.
Yes.
This C, what does it represent?
What?
C is
Here it's all the C's. It's it's the vector. So you have all the vectors. It's like all the concentrations, right? So if you want, it's in some sense it's a way to recapitulate this, right? So I'm sort of ideally, I mean, if you want to, we don't go there. But in principle, you could imagine that each of these I is a node in this plot, right? And the amount you have here depends on this one because you have a reaction, or let's find one which is more readable. So for instance, the amount you have here, right? Depends on the amount you have here, depends on the amount of this one. but also depends on the amount of this one because this blocks the reaction. Okay? Okay, good. So what I hope I convince you is that in our journey to looking inside cells and see what they do, we want to be in exponential phase. And actually, this is something that it's less standard than what you could think, but the idea is if you ever want to do an experiment, should really make sure that you are in exponential. I mean, let's say it depends on the experiment, but the kind of experiments that I'm going to show you obtain a clean result exactly because they made sure to be in exponential phase. So actually what you do practically is that you take cells, you grow them once, you see where this point here, you take again cells, you grow them to this point, then you take them and you give fresh food. And then if you do that, they start growing exponentially, and they grow exponentially for a long time, and then you can really measure things. Okay? So you want really to be sure that you are in this phase here because you know that there is a fixed point. I mean, this is the idea. Questions?
This fixed point is stable.
Okay, so regarding this question, what we can do is to use this setting to study a simple example, going back to arginine. I can perhaps just tell you what the idea, and then you can sort of do it. I don't know if as an homework. I mean, you can do it by yourself because it's pretty simple. So let's think of a system on this kind of scenario, which is analog to the arginine system we described. So okay. Okay. So to this one. So basically, what we want to describe here is the dynamics, sort of written in this way, of this complex of these proteins and the production of arginine. Okay, and show you how these dynamics here. So now we are not necessarily coupling it with growth, but we want to show how these dynamics here produce regulation. Okay, that sort of is the same mathematical set. So I'll just tell you the setting, and then you can do it. So imagine that we want to write the dynamics for the concentration of P, and P is whatever protein is produced here, is the protein that then allowed to synthesize arginine. Okay? So in general, what we can imagine is that it is produced at some constant rate. I mean, the simplest case, and then it's diluted depending on growth. Okay? Now, this production here, okay, well, this is, let's say, one situation. Then I'll make it more complicated. And then what you can imagine is that you have the production of arginine, which depends on some substrate times P. Okay? And then because of growth, this is also diluted. Okay? Yes? Yes, this is like enzymes that, it's like characterize the enzymes that catalyze the reaction that contribute to the biosynthesis of arginine. So P is what makes, allow you to produce arginine. Okay? So basically you have S that becomes arginine, some substrate, and P is the enzyme that catalyzes this reaction. Okay? This is the simplest set. Okay? Now the idea is this is like completely unregulated, right? So here, if you do this, you always produce, what happens is that you always produce a fraction alpha, right? And then it gets diluted. And this rate here, well, you go to steady state, and so you always produce a fixed amount. You always produce a fixed amount P, okay? What I want to show you is the effect of regulation, okay? The fact that you can regulate and have a feedback on how much you produced based on alpha, okay? And so I'll set up the setting, and then you can do it by yourself, okay? So the okay. So what determines okay, what determines the production of our enzyme, okay, is whether this repressor here, so this site here, has a repressor that is bound or not, okay? So if it's bound, so B represents whether it's bound or not. If it's bound, you don't produce it. If it's unbound, you produce it, okay? So you have a rate at which you go from bound to unbound, okay? And the rate at which you do the opposite, okay? So the rate at which you go from unbound to bound, it's something that depends on the presence of arginine. Okay. And now for simplicity, we can assume that this rate is independent. Okay. So if there is arginine, you bind, but then when you're bound, you can unbind it like with a constant rate. Okay. Good. So this means that if you take this two-state system, you can measure, you can look at how right, no, sorry, no. So this is proportional to the number of regulators that have the, sorry. This is proportional to the number of regulators that have the arginine bound, okay? And so if you look at what is the fraction B, this is equal to the regulators that are bound divided by, well, this is right, KB, KB RA plus K unbound, okay? And then you have to decide how a regulator becomes bound, becomes active or not, and that depends on arginine, okay? So again, what you have is that regulators can be active, well, can be, let's say, active if there is arginine bound, or inactive, and this is proportional to some gamma times the presence of arginine, and this is the rate at which the arginine unbinds, okay? And so again, you have a two-state system, and you have that the two rates, so you have a total number of regulators, and the active regulators times gamma unbound is equal to gamma bound times arginine R minus RA. Okay? And so here what you get is that RA is equal to gamma B times R times A over A gamma B plus gamma U. Okay? And if you put these things together, what you get is that this bound is something which is equal to some B max 1 plus 1 over A over AC. Okay? Where this A and the AC depends on all these parameters. Okay? So basically here you have something where this depends on A. Yes? Here? So what you have here is that this is the rate, so you have this regulator which can be bound to DNA. So this is bound or unbound, okay? And these applies only to the ones which have arginine, okay? And this one, it's the rate at which you have something without arginine or you have something with arginine, okay? So you have these two processes. Okay? So in order to be bound, you need to have arginine. And so you have this process here, and you have this process here. Yes?
Every time you write, you pass from this reaction formally to this expression, you're assuming a sort of stationary.
Yes, I'm assuming that that is fast.
Okay, so there is some sort of separation of time.
Yes, yes, yes. Okay? Yes?
Sorry, I thought like in the second case where active and inactive we are talking about, there only the arginine concentration matters. And in the first case, where it's binding and unbinding, I mean there it's independent of it. It depends on the receptors.
No, this is only, it applies only to the ones with arginine bound. Okay? Okay?
You can have arginine and be unbound.
You can have, if you have arginine, you are always unbound. If you don't have arginine, yes, you can have arginine and be unbound. There is a rate, right? Right. So the point is that now you have a dynamical system when you can basically respond to, I mean, where you have a coupling between the production rate and the presence of arginine. Okay? So the question for you is that you can study this, so you can study the fixed point, and you can study how this system responds when you, for instance, change the substrate. Right? So suppose that this is equal to 0, and then you put it different from 0. Okay? So you can make arginine. How does the system respond to that? Okay? Yes? Yeah, I know. You should have killed me. Okay, if you want, this is active, this is inactive, and I can say this is active, active, active. Okay. Okay.
VS represent what?
This is like it's how much substrate there is. Okay, you want to make arginine, it's how much substrate there is. Okay. So this can be, I mean, you can imagine that this is a parameter, right? But the idea is that, for instance, you can study the case where S is absent and then you produce it, or when it's present and then gets depleted, right? So you can study the response of this when this disappears.
It's like the glucose in the system.
Yeah, I mean, it's like, yes, something. Okay. Questions?
So we can assume that VS is constant.
If you can assume that Vs is constant and you find a solution, a fixed point solution, okay? Then you can say, suppose that this varies, and you can imagine that you have perturbations, you remove all the arginine, or you add the substrate, you can look how the system responds when you have this B. Okay? Yes? No, this is like the amount, it's the rate. Yeah, it's basically, no, this, you can imagine that this is like some parameter V max times S over S plus Km. Okay? Questions? Okay. So you can play with these, and then we can discuss. Well, let's see it at the end. Blah 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 blah. Okay. Good. So, okay, but in general, let's say we are in this setting here, and so we can imagine that if we open a cell, we have all these molecular species going around. Okay. And so the first question we can ask is, okay, let's start to build some coarse categories, okay? Some very coarse categories, and measure how much of these coarse categories do we have, okay? And so people started, for instance, take E. coli in exponential growth and start measuring all the main classes of materials that make up a cell, okay? So for instance, what you can do is, you can say, suppose that I take a cell, I remove water, okay? And they start measuring how much, let's look at the first column, how much of the weight is protein, okay? 55% is protein. How much RNA? 20%. How much DNA is 3%? How much lipids? 9%. And blah, 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 blah. Okay? So you can do that. And, okay, you can imagine that you can start making tables of these all the time. I mean, you can, this is like E. coli, a particular strain, growing at 37 degrees. In aerobic glucose, minimal medium, so it means with minimal salts that are needed to grow, glucose as a main carbon source, and with oxygen, okay? Very good. Then you can say, okay, I do another condition, I'll find another table, okay? So the first question that we can ask is, if we take this very coarse perspective, okay, looking how much protein you have, how much DNA you have, how much RNA you have, how does this depend on the conditions, okay? Now, when you measure these, the first question that you can ask is, okay, I can measure proteins, I can measure RNA. I mean, the main two categories are proteins and RNA. What I'm actually measuring, okay? So these are the two main. So what are you actually measuring when you measure protein and RNA? Well, the first thing you can do is, okay, we can measure these things. Now, we understand this argument here, so if we want to compare condition, we want to measure this, in what condition should we measure it? In exponential growth, right? So we say, okay, now we make sure that we are in 37 degrees aerobic glucose minimal medium, and we are in exponential growth. We extract the proteins, we extract RNA, we weight them, and we look, I mean, for instance, what is the ratio, or we look at how much we have, okay? The second question is, no, we do that, okay? And we repeat for another condition, we repeat for different conditions. So, yes? Yes, so we do what I mentioned earlier. So what you do is that you do an experiment where you make sure that you are in exponential growth, okay? So let's say you consider a condition, you consider glucose, okay? And you grow them in glucose for a long time, to simplify, until you are sure that they are in exponential growth and they have been in exponential growth for a while, so then you're sure that this condition of stationarity is met. Okay, so is the question clear? What we are trying to do is we have, let's say, these two categories here, and we want to measure how big these two categories are as a function of the conditions. Okay? Now, I open a disclaimer because it's important. So in explaining what I am about to explain, there are two possible ways, two possible routes. One is to show you the data and build an understanding out of the data. Right? So you say, I showed you one experiment, we try to understand one experiment, and then we say, oh, this is another experiment you can do, this is what we understand about the theory, we build piece by piece, and we move on this. This will be one way. The other way will be, I tell you what the model is, and then we test it, and it works perfectly. Okay? These are the two ways. We're going to do both. So I'm going to start with the first one. So we're going to look experiment by experiment. We're going to sort of construct the pieces. At a certain point, I'll stop, and I'll tell you, okay, we have understood this. Let's write down some equations. Let's derive predictions. Of course, we will be able to recapitulate what we have found. Everything will work, and we will be very happy. Okay? Good. Close parenthesis so you know how we're going to proceed. Now, this is like historically what people did, right? Okay, we want to understand what the E. coli is doing. We start measuring stuff inside E. coli. The main two stuff to measure are proteins and RNA. Now, I don't care about the total weight, right? Because the total weight, this is a colony growing. If I take twice as many cells, I have twice the weight. So I care about proportions, okay? So one simple thing I can do is to measure how much RNA do I have per protein, right? So proteins are the majority of the weight. RNA is the second category. How much RNA I have per protein, okay? And so people did, okay, so I have this measure. And then I want to somehow relate it to conditions, okay? Let's say that I consider different sugars. Okay? How do I change condition? I could have billions of choice, right? But one simple condition is to say, I'll change the nutrient. I give glucose, I can give lactose, I can give mannose, I can give maltose, I can give many things that finish in O's because they're sugars. Okay, I have all these things, right? And I have many tables. I have one table for glucose, one table for mannose, one table for maltose, one table, etc. Right? What do I do with this table? Yes?
Can you measure like the actual amount of RNA related to the production of a certain protein?
You can do it.
Okay.
You can measure messenger. So here it's all the RNA that is in the cell. Not all the RNA is messenger RNA. Actually, the messenger RNA is a small fraction of the total RNA. I'll go through that later. But it's all the RNA that is in a cell. Okay? But you can do, I mean, one option of what to do is exactly to, I mean, you can do experiments where you measure the mRNA related to a particular protein. Okay? But now we are sort of starting from the beginning. We are starting from the 60s. Okay? So you measure these two things, right? And so we have many of these tables. Now you want to make a plot. Right? So on the y-axis, we can measure, for instance, RNA to protein. How much RNA over proteins you have. Okay? What do we put on the x-axis? We have many conditions. Well, I can do a plot where I say RNA, let's say, to protein. And I could say, here I have glucose. Here I have mannose. Here I have maltose. Here I have lactose. And then I'll get, sorry, I'm thinking what comes. I have this. What do I learn? This is not a nice way to plot it. So what would you put on the x-axis?
Maybe the growth rate.
Good idea. Maybe the growth rate, right? So I put the growth rate, which is basically telling me how fast I go in that condition. Okay? So now, when you do this, I mean, you have not been, most of you have not been exposed to biology, but when you do this kind of plots, if you have this thing in mind, your expectation is that the plot looks like, if you are lucky, the plot looks like this. I mean, if you have few points, you have, okay? So you say there is a correlation, a significant correlation. This is like the baseline of the plots you expect, okay? Just to tune your expectation. So when people did this in the 60s, what they find is something much more clean, okay? So actually, this is the plot with the reverse axis. I will go back to the initial one. And so what you do is that here you have RNA to protein on the x-axis and growth rate on the y-axis. And there you have different nutrients, okay? So you have, for instance, 8 is glucose, okay? 10 is, I don't remember, but it's something plus the addition of amino acids. So when you add amino acids, cells grow faster because they don't have to produce them. Glycerol plus amino acids, etc. So you have different conditions. And what you see is that if the growth is low, you tend to go to a plateau of this ratio, okay? And when growth becomes faster, you tend to have something that is more linear, okay? Now, slow growth, it's always hard to investigate because imagine you have to go to well, slow growth is harder to investigate. People repeated the plot 50 years after, only for fast growth, and now the axes are reversed. So it's RNA to protein versus the growth rate, as I showed you there, okay? And here you have those points repeated, so it's 5, 6, 7, 8, 9, 10. And the new conditions. So you have M63, which is like a minimal medium, so it's just salt, plus glycerol and glucose. Then cas amino acids, which is a type of amino acid, plus glycerol and glucose, and rich medium, so many amino acids plus glycerol and glucose. Okay? Yes.
Is there significant differences in the composition of a cell between like when it's divided, like when it's just born and when it's like
yes. So this is, you're going to look at this in 2 weeks, individual cells. The point is, there is a difference because there are fluctuations. Now, what I'm saying now is, if you look at as a first approximation, if you look at a colony, you can neglect these, because if cells are not synchronized, at the colony level, you will have cells that are born big, cells that are born now, cells that were born earlier, so they are at any stage of the life cycle. So I'm writing a model for whatever stage of the life cycle of a cell, from growth to division. Yes.
And in the lifetime of the cell, what percentage of it is mitosis?
So this is, you're going to see, yeah, I mean, I can tell you, but it's a detour. So let's say what a cell has to do to divide is has to start replicating DNA, and then create a septum and divides. And it depends on the condition. So actually, if you are in fast condition, you start replicating, a cell starts replicating the DNA, not just for the daughter, but for the granddaughter or the grand-granddaughter. Yeah. So it's a whole story. For now, for us, if you look at concentrations, it's not precisely true, but you can sort of neglect the fact that a cell divides. Right? Because if a cell divides in 2 and it's equal, on average, the concentrations are fixed. Okay? It's not precisely true, but let's say. Okay? So the first observation that we have, so this is, first of all, this is a surprising plot. Okay? Now, you are not very surprised because you are spoiled by plots which work perfectly, but this is a surprising plot. Because what you have here behind is all that complex network of reactions, of regulation, etc. And here you have different conditions, right? You have different conditions, so it means that the kind of environment you consider, it's very different. You have very different inputs, right? So it's like if I told you, you start from here, right? And you give this thing here as a food, okay? Or you give that thing over there, okay? Or you give that thing over there plus amino acids which are somewhere else, okay? Then, yeah, of course, you will have some dynamics. Yes, you are in exponential growth, so you go to some steady state, etc. But why you should expect that then somehow the composition measured by RNA to protein becomes sort of so, in a relation so nice with the growth rate, it's not obvious at all. Okay? Yes?
I have a question. Just want to make sure that I understand the plot.
Yeah.
So here we're seeing different initial conditions. So like if we use glucose as a source of nutrient, this is the growth rate that we get from the exponential phase and the duration.
Yes. So basically, imagine that I want to do the 2 experiments, right? For 2 points. So I have a flask where I put glucose. I put E. coli inside. I have it growing in a way such that I'm sure that is in exponential growth. So actually what you do is that you grow, you dilute, etc. But you are sure that you are in exponential growth. So let's say that they lag and then they are in exponential growth and then they saturate. So you are here, okay? So you measure the slope. This is the growth rate lambda. okay? And then you take cells here from the flask here, and you extract RNA, and you extract proteins. You weight them, you take the ratio, okay? Then you have another condition. Let's say glucose plus amino acids, okay? Do the same. Now, when they grow here, they go much faster, right? So the slope is bigger. This is log biomass. This is log biomass, okay? So they grow faster. So you take it here, you take the sample, and you measure the two quantities. Yes?
So the ratio of RNA over proteins kind of, is it proportional to the rate of catalysis? Or?
This is the last point I wanted to make today, and then we stop, okay? Other question before I go to the stop? Of course, now no one wants to ask questions. So, okay, we have this RNA protein. What does RNA protein actually measure? Okay, so proteins, it's whatever does something in the cell. The question is RNA is in many parts, as was asked. So it's in messenger RNA, it's in tRNA, transfer RNA. Most of RNA, so here you have the different RNA categories. So you see that you have these 23S rRNA, you have all these rRNA, and then you have transfer and messenger. So the total amount is 58 grams to the, whatever measure is this one, which looks odd. So it's whatever unit of measure is this 58. The majority, so 48 of this 58, is in these rRNA, which are the RNA structural component of ribosomes. So ribosomes, those machines that makes protein, that assemble proteins out of messenger RNA, that reads the messenger RNA, translate the messenger RNA to a sequence of amino acids, those big chunky proteins are also made of RNA. Okay, they have a structural component that is made of RNA. And this structural component is what makes most of RNA of a cell. Okay? So when you are measuring RNA over protein, effectively what you are measuring is how much ribosomes you have. What is the fraction of, with some unit conversion that we will do next time, but you are effectively measure what is the fraction of mass that is ribosomes. Okay? Good. So plan for next time. So play with the can we answer the question next time? Okay? Because, there are two ways to answer this question. One question is, one way to answer this question is sort of a priori, which is ribosomes are, we care about ribosomes because the ribosomes performs essential functions in a cell, which is true for anything that is inside a cell. Okay? I will answer at the end of next time, because otherwise, yes, it's better. Okay? Thank you. See you tomorrow.